The idea that major psychiatric disorders arise from an excess or deficiency of a single neurotransmitter like serotonin, dopamine, or norepinephrine has roots in a simpler era of psychiatry. This belief gained traction when early medications produced dramatic changes in mood and behavior. A single pill could lift someone from catatonic depression or dampen the intensity of manic episodes. And it seemed, for a moment, that mental illness could be mapped neatly onto the brain's chemistry. The logic was seductive. If a drug altered the mind, then the mind must be broken in some precise, chemical way. This was more than a medical observation. It became a cultural myth, a narrative that filtered into public understanding. Depression was framed as a serotonin problem, and anxiety as an imbalance in the brain. It was simple, digestible, and reassuring, but reality, as it so often is far more complex. Mental illness does not exist in a vacuum of molecules. It arises at the intersection of biology, environment, trauma, thought, memory, identity, and society. The brain is not merely a chemical soup. It is a dynamic, adaptive organ shaped by experience, relationships, and time. And while medications can play an important role, the idea that they are correcting a precise imbalance is more metaphor than science. This isn't to say that neurochemistry is irrelevant, far from it, but the notion of a single faulty neurotransmitter as the root cause of deep psychological suffering oversimplifies the intricate dance of systems that define the human mind. There is a crucial distinction between how chemicals interact with the brain and how they circulate through the rest of the body. While the body as a whole is relatively accessible to substances carried in the bloodstream, the brain is guarded by a unique and highly selective security system, a biological fortress known as the blood-brain barrier. This barrier isn't a wall in the traditional sense, but rather a finely tuned filtering system made up of tightly packed endothelial cells lining the brain's blood vessels. It serves one vital purpose: to protect the brain from potentially harmful invaders, bacteria, toxins, or even certain immune cells that might otherwise freely enter other tissues of the body. In most parts of the body, when a threat is detected, whether it's a virus, a parasite, or a foreign chemical, the immune system launches an immediate and often aggressive response. White blood cells surge toward the intruder. Inflammation rises, and a full biological defense is activated, but the brain plays by different rules. Here, the immune system is restrained. The brain cannot afford the same kind of inflammation that might occur in a swollen knee or infected cut, because even slight swelling in this enclosed, delicate space can be catastrophic. So evolution built a checkpoint. Only specific molecules, nutrients, and gases are permitted to cross, and only under the right circumstances. This means that even if a drug is circulating through your bloodstream, it doesn't guarantee access to your brain. Many pharmaceuticals must be specially designed to bypass or penetrate this barrier, and even then, their effects are shaped by how the brain absorbs and responds to them. In essence, the brain is both a master controller and a protected sanctuary. It is simultaneously central to everything we are and fiercely defended against everything we are not. Understanding this barrier is key to understanding why treating mental illness is not as simple as delivering chemicals to the brain. The route in is narrow, the rules are strict, and the consequences of getting it wrong can be profound. The immune system is a marvel of biological engineering, a layered defense network that detects, attacks, and removes foreign invaders. It operates with a complex set of tools: antibodies that flag threats, white blood cells that engulf enemies, and chemical messengers that coordinate the battle. But among its most primitive and most dangerous tactics is a blunt force response, killing infected cells outright. This method works well enough in many parts of the body. If skin cells become infected, the immune system may destroy them, allowing healthy tissue to regenerate. Even organs, like the liver or lungs, can often withstand this kind of cellular loss and repair themselves over time. The cost is acceptable, because the body can recover, but the brain is a different story. Neurons, the primary cells of the brain, are not easily replaced. They do not divide and regenerate like skin or blood cells. Once lost, they are often gone for good. So imagine, for a moment, if a bacterial colony were to invade a part of your brain. If the immune system responded with its usual scorched earth policy, destroy the infected to protect the whole, the result would be catastrophic. You wouldn't just be eliminating a threat, you'd be erasing parts of your memory, your ability to think, your capacity to feel, reason, or speak. That's why the brain's immune strategy is uniquely cautious. It cannot afford collateral damage, it cannot risk widespread inflammation or indiscriminate destruction. So instead of deploying an army of killer cells, the brain relies on microglia, specialized immune cells that act more like careful custodians than soldiers. They monitor the environment, respond selectively, and only act when absolutely necessary. But this delicate balance has its price. It means the brain is both protected and vulnerable, shielded from overly aggressive immune responses, but also slower to detect and respond to certain infections. It's a calculated trade-off, cognitive preservation over brute survival. This is the paradox of the brain's defense. Too little immune activity, and harmful agents may thrive undetected. Too much, and the very structures that make you, your thoughts, memories, and consciousness, might be collateral damage in the body's war against itself. To protect the most vital organ in our body, the command center of thought, memory, and self, evolution crafted a gatekeeper, the blood-brain barrier. Unlike other parts of the body where blood freely delivers nutrients, hormones, and immune cells to tissues, the brain is wrapped in a far more selective security system, one designed not for speed, but for precision and protection. This defense is not metaphorical, it is literal, a tightly woven wall of endothelial cells lines the capillaries that thread through the brain. These cells are fused together by what are known as tight junctions, forming a near-impermeable seal that prevents substances in the bloodstream from leaking into the brain's fragile neural tissue. This barrier is discerning tiny essential molecules such as oxygen, carbon dioxide, glucose, and certain hormones are granted access. They pass through specialized transport systems or diffuse across membranes, nourishing the neurons and keeping the mind awake and alive. But for most toxins, pathogens, and foreign chemicals, the answer is simple: do not enter. Even many medications designed to help the body, antibiotics, chemotherapeutic agents, or pain relievers, are turned away at this cerebral gate. The blood-brain barrier does not care about your intentions. It only allows what it deems safe, often erring on the side of caution. This system works beautifully. Most of the time, it keeps out bacteria that could cause meningitis. It filters away toxins that might disrupt our neurotransmitters, and it dampens immune responses that would otherwise turn our own cells into collateral damage. But there's a cost to this kind of protection. When trouble does breach the barrier through trauma, inflammation, or infection, the brain's natural defenses are limited. The very gate that shields it from harm also limits the tools it can use to heal. It's a paradox of the highest order. In order to keep the brain safe, evolution wrapped it in a vault, but even vaults can be breached, and once they are, the response must be delicate or devastating. This finely tuned barrier, so masterfully designed to protect, also comes with a consequence. It keeps the brain safe. Yes, so safe, in fact, that bacterial infections of the brain are astonishingly rare. The very diseases that might devastate the lungs or liver often fail to touch the sanctity of the mind, simply because they cannot get in. But this same barrier that defends us also defies us. For those who seek to heal the brain, scientists, doctors, and pharmaceutical pioneers, the blood-brain barrier presents a daunting obstacle. You cannot simply swallow a pill and assume its contents will reach your thoughts, no matter how potent a drug may be, no matter how carefully engineered its chemistry. If it cannot pass through the barrier, it cannot reach the neurons, it cannot do its work. The blood-brain barrier is selective to the point of stubbornness. It resists intruders with no understanding of intent, turning away both the poison and the potential cure. And so, for pharmaceutical companies, this becomes a battlefield of a different kind, not one of disease, but of design. They must find clever molecular keys to unlock the gates, designing drugs that are small enough or lipophilic enough or masked in such a way that they slip past the watchful guards unnoticed. It is not enough to know what needs to change inside the brain. One must also find a way in, a way past the sacred veil that evolution built to ensure our most precious organ remains untouched, uninfected, and unreachable. This is why so many treatments for neurological and psychiatric conditions remain limited, not because we don't know what's wrong, but because we cannot yet whisper the cure through the wall. Yet not all is kept out. Some molecules, by design or by accident, possess the right characteristics, the right size, the right shape, the right chemistry to slip quietly past the blood-brain barrier and into the sanctum of the mind. Caffeine, for example, is one such quiet infiltrator. It passes through with ease. Not because it forces its way, but because it wears a disguise. Caffeine mimics adenosine, a naturally occurring molecule found throughout the nervous system, a kind of molecular lullaby that tells your brain it's time to rest. But when caffeine takes its place, it blocks that message. It doesn't stimulate you directly. It simply silences the messenger of fatigue, and in doing so, the world feels a little sharper, your mind a little brighter, though nothing truly changed, except your access to sleep. Alcohol, too, drifts through. Its molecular structure and electrochemical properties make it easily absorbed, not just into the bloodstream, but through the blood-brain barrier itself. It crosses with little resistance. And once inside, it begins to alter perception, slow reflexes, and disinhibit the parts of us we usually keep guarded. It's not a polite guest. It enters, uninvited, and rearranges the furniture. Fortunately, some psychiatric medications have also found a way in. Many antidepressants, antipsychotics, and mood stabilizers are made with small, fat-soluble compounds, tiny enough, slippery enough to pass the barrier's gates. These molecules are designed to mimic or modulate the brain's own neurotransmitters, serotonin, dopamine, norepinephrine. When they work, they can be life-altering, but not all drugs are so fortunate. Not all molecules are small enough, not all chemistry can be made to conform, and so, for every compound that crosses successfully, there are dozens that are denied entry, promising in theory, powerless in practice. The blood-brain barrier is not cruel, only careful. It guards what it was built to protect, but in doing so, it makes healing the mind one of the greatest scientific challenges of all. Along the twisting corridors of the blood-brain barrier, not all doors are sealed. Hidden within this vigilant wall are specialized gates, molecular passageways designed with intention, evolved with precision. These gates are not generous, but they are discerning. They allow passage to specific larger compounds, nutrients the brain cannot produce on its own, but desperately needs: glucose for energy, amino acids for neurotransmitter synthesis, and certain hormones that guide growth, emotion, and metabolism. Each gate operates like a sentry with a keyhole shaped for only one kind of key. Insulin, for example, is admitted not because it is small, but because the brain has designed a precise receptor to recognize it. Leptin, the hormone of satiety, can cross, but only under tightly regulated conditions. Even iron and vitamins, in their proper form, are permitted safe conduct through their designated channels, but for most pharmaceutical compounds, the story is very different. No matter how powerful a drug may be in theory, no matter how well it works in a petri dish or a bloodstream, if it cannot pass through this barrier, it might as well not exist at all. At least to the brain, the molecule arrives at the gate, waits, and is quietly turned away. It's not enough to design a drug that works. Scientists must also design a molecular passport for it, one that matches the structure, solubility, and molecular behavior that the barrier will tolerate. This is one of the great frustrations of neuroscience. The cure might already exist, but the brain won't let it in, and so, behind this wall of molecular silence, the brain continues its work untouched by most of the modern world. For better and for worse, but getting through the blood-brain barrier is only the first trial. Once a compound finally crosses into the brain's protected interior, the real work begins. It must do more than arrive. It must interact, intervene, and ultimately influence how the brain functions without disrupting the delicate symphony that is always already playing. This is where the science turns from chemistry to choreography, because the brain doesn't run on random signals. It runs on neurotransmitters, the elegant chemical messengers that allow neurons to communicate, coordinate, and create everything from movement to memory, emotion to identity. Each neurotransmitter, serotonin, dopamine, GABA, glutamate, norepinephrine, and more, operates like a note in a score. They don't just float freely, they are released, received, recycled, precisely timed and exquisitely placed. They move through synaptic clefts, binding to receptors like keys fitting into locks, sparking electrical activity that either excites or calms a neuron's next move. For a drug to work, It must join this conversation, and not as a brute force interruption, but as a subtle participant. It may need to mimic a natural neurotransmitter, fitting into a receptor as if it belonged there, or it might block a reuptake channel, prolonging a signal's presence, as many antidepressants do. Some compounds alter how neurons release their messages. Others change how the receptors respond to them, but every intervention carries a consequence. Tweak one system, and others shift. A slight boost in serotonin might lift a mood, but also disrupt sleep or appetite. An increase in dopamine may relieve depression or trigger anxiety, restlessness, or compulsive behavior. That's why neuroscientists focus so heavily on the role of neurotransmitters, because they are not just chemicals, they are the brain's language, and to influence the mind, with any hope of healing, we must learn to speak it fluently. Neurons, the mysterious messengers of the brain, speak in electricity and chemistry, in silence and sparks, and like all good conversations, their structure shapes their message. At one end of the neuron are the dendrites' delicate, branching filaments that stretch out like the arms of a tree, reaching for connection. These are the receivers, the listeners, the places where incoming signals from other neurons arrive. Each dendrite is studied with receptor sites, tuned to pick up the subtle whispers of neurotransmitters flowing across synapses. At the other end lies the axon, a slender, elongated extension, often stretching long distances through the brain or body. This is the transmitter, the carrier of the message, sending the neuron's internal decision onward to its next destination. Both dendrites and axons are not passive wires, they are alive with gates, specialized proteins embedded in their membranes called ion channels. These gates do not open for just anything. They are highly selective, only allowing specific charged particles, like sodium, potassium, calcium, or chloride, to pass through. The opening and closing of these channels isn't random. They respond to changes in electrical charge, chemical signals, or even mechanical pressure, and when the right gates open in the right sequence, a ripple of electrical energy, known as an action potential, races down the axon like lightning along a cable. This traveling signal is what allows one neuron to send its message to the next, and it all begins with the selective permeability of these tiny, invisible gates. They are the guardians of neural communication, deciding what comes in, what stays out, and when the next thought, feeling, or movement will be born. Along the delicate branches of the dendrites, the gates take on a special role. Here, they are known as receptors, so named because they are built to receive not just anything, but very specific molecules, neurotransmitters, the chemical messengers that carry signals from one neuron to the next. Each receptor is finely tuned, almost like a lock waiting for a perfectly shaped key. Only a particular neurotransmitter, whether it's serotonin, dopamine, glutamate, or GABA, will fit just right and trigger a response, but what happens next depends on the nature of that chemical message. Some neurotransmitters excite the neuron, urging it toward action. Others inhibit, calming the electrical storm and preventing a signal from firing. One of the most common effects is this: when a neurotransmitter binds to its matching receptor, it can cause that receptor to open a gated channel embedded in the cell membrane. Through this channel, charged ions such as sodium, potassium, or chloride flow in or out of the neuron. This flow of ions alters the delicate balance of electrical charge inside the neuron. Neurons maintain a careful distribution of positive and negative ions. It's part of how they prepare to fire, so when a receptor opens a gate and lets these ions shift, it can tip the internal voltage closer to or further from the threshold needed to spark an electrical impulse. In this way, neurotransmitters don't just deliver a message, they reshape the electrical landscape of the neuron itself. They are the unseen hands adjusting the inner current of thought, emotion, and memory one molecule at a time inside every neuron. There exists a fragile tension, a carefully maintained electrical balance. The interior of the cell holds a slight negative charge compared to its surroundings. Outside, the extracellular fluid brims with positive ions, like sodium, waiting at the gates. This difference in charge creates potential, not just electrical, but biological. It's as if the neuron is holding its breath, its gates shut tight, keeping the flood at bay. But everything changes when the neuron receives a signal. When neurotransmitters bind to receptors on the dendrites, certain ion channels, specialized gates, begin to open. With this opening, positive ions that had been yearning to enter now rush in. driven by both electrical attraction and chemical gradient. As these positive charges flow into the cell, the inside of the neuron grows less negative, inching toward neutrality, and then, if enough gates open, it tips past a critical point. The threshold, this moment is everything. Crossing the threshold triggers an action potential, a powerful, self-propagating wave of electrical activity. Like a spark catching fire, the charge surges down the neuron's axon, a long, thin projection that carries the message forward. The action potential doesn't fade as it travels, it's all or nothing. Igniting gate after gate in sequence like falling dominoes, racing toward the axon terminals at incredible speed. And when it arrives at the end of the axon, it doesn't stop. The electrical signal triggers the release of neurotransmitters into the tiny gap, the synapse, between neurons. From there, the message leaps across space to the next cell, repeating the cycle again, neuron to neuron, spreading through the vast, intricate circuitry of the brain. Each signal, each threshold crossed, is a whisper in the endless electrical symphony that makes us who we are. Our thoughts, our feelings, our memories, all born from the silent longing of ions to find their way home. Not all neurotransmitters encourage a neuron to fire. In fact, some do the opposite. While certain neurotransmitters act like green lights, opening gates and allowing positive ions to flood in, increasing the chance of an action potential, others are more like red lights, sending a message of restraint. These inhibitory neurotransmitters block or close I don't channels, or they open gates that allow negative ions such as chloride to enter the neuron. This makes the inside of the cell even more negative, moving it farther from the threshold and less likely to fire. In essence, these neurotransmitters don't silence the neuron entirely. They simply make it harder for the neuron to speak, and just as there are different messages, there are different messengers and listeners. Neurotransmitters don't bind randomly, they interact with specific receptors, like a key fitting into a lock, but each neurotransmitter can often bind to multiple types of receptors, and each receptor type can trigger different effects inside the cell. For example, the same neurotransmitter, say dopamine, might excite one neuron while inhibiting another, depending entirely on the type of receptor it binds to. This variety allows the brain to work with stunning complexity. It's not a simple on or off switch, it's a vast network of subtle influences, a symphony of excitation and inhibition, shaping every thought, emotion, and movement we experience. The language of the brain is chemical, but the meaning comes from the context from which neurotransmitters are released, where they land, and how the receiving cell chooses to respond. Schizophrenia. The word schizophrenia is one of the most misunderstood and misused terms in our everyday language. In casual conversation, people sometimes describe someone as schizophrenic when they act inconsistently or seem to display conflicting behaviors, as if the word meant split personality or a fractured self. You might hear someone say, "He's being so schizophrenic today," to describe mood swings, unpredictability, or a sudden change in attitude. But this popular usage is not only misleading, it's also deeply inaccurate. Schizophrenia is not the same as having a split personality. That misconception likely stems from a misunderstanding of the word's Greek roots, schizo-split and phren-mind, which refer not to multiple identities, but to a fragmentation of thought, perception, and emotional responsiveness. It's a serious psychiatric disorder, often marked by hallucinations, delusions, disorganized thinking, and difficulties in distinguishing what is real from what is not. Misusing the term to describe erratic or contradictory behavior not only spreads misinformation, it also contributes to the stigma surrounding mental illness. It turns a complex and often debilitating condition into a careless metaphor. And in doing so, it risks minimizing the lived reality of those who struggle with it. For individuals diagnosed with schizophrenia, the condition is not a quirky personality trait. It is a daily challenge that affects their perception, communication, relationships, and functioning in the world. Dismissing or distorting this reality through offhanded remarks adds to the barriers they already face, from misunderstanding and fear to social exclusion. To speak more compassionately and more accurately, we must be careful with the language we use. Words carry power, and when we use them thoughtfully, we help create a culture of awareness, respect, and dignity for those navigating the complexities of mental health. The term schizophrenia, was originally introduced in the early 20th century by Swiss psychiatrist Eugen Bleuler, who coined it from the Greek words schizo to split and phren mind, but what he meant by split mind was not a split personality, rather, he was referring to the fragmentation of thought processes, emotional disruption, and the disconnection between thoughts, feelings, and reality that characterize the disorder. In essence, schizophrenia was named to reflect the disordered, often chaotic nature of cognition in individuals with the condition. Their thinking can become fragmented, illogical, or detached from reality, but not in the way commonly misrepresented in pop culture. Importantly, schizophrenia is an extremely heterogeneous disorder. This means that two individuals who share the same diagnosis may experience entirely different symptoms, severities, and life impacts. One person might struggle with hallucinations and delusional beliefs, while another may present more with disorganized speech, emotional flatness, or cognitive difficulties such as trouble focusing or remembering. In some cases, symptoms are more positive, meaning they involve the presence of abnormal experiences like hearing voices or believing in conspiracies. In other cases, negative symptoms dominate, such as withdrawal from social life, lack of motivation, or diminished expression of emotions. This diversity in presentation makes schizophrenia both challenging to diagnose and to treat. It is not a one-size-fits-all condition. Instead, it exists on a broad spectrum, and clinicians must often tailor their approach to each individual's unique profile, needs, and lived experience. Understanding this complexity is essential. It reminds us that behind the label of schizophrenia is not a stereotype, but a person navigating a uniquely difficult and deeply personal experience of the world. Two individuals diagnosed with schizophrenia may exhibit vastly different clinical profiles, highlighting the disorder's profound variability and complexity. For example, one patient may primarily experience what are known as positive symptoms. In psychiatry, the term positive doesn't imply something beneficial. Instead, it refers to the presence or exaggeration of abnormal behaviors or experiences that are not typically seen in healthy individuals. These can include delusions, firmly held false beliefs, often paranoid or grandiose in nature, hallucinations, most commonly auditory, such as hearing voices, disorganized thinking, and erratic or unpredictable speech or behavior. These symptoms represent an increase in psychological activity. The mind becomes overactive, sometimes chaotic, creating a distorted or altered experience of reality. In contrast, another patient might show a predominance of negative symptoms, which are equally debilitating but often less visible or misunderstood. Negative symptoms refer to the absence or reduction of normal mental functions or behaviors. These might include flattened affect, reduced emotional expression, alogia, poverty of speech, anhedonia, inability to feel pleasure, social withdrawal, and even catatonia, a state of motor immobility and unresponsiveness. In these cases, the patient may appear emotionally distant, unmotivated, or disconnected from the world around them, not because they don't care, but because the disorder has diminished their capacity to engage. This spectrum of symptoms can affect every aspect of a person's life, from relationships and communication to work, self-care, and identity. It's also possible for someone with schizophrenia to experience both positive and negative symptoms at different times or simultaneously, which further complicates treatment and diagnosis. Recognizing the difference between these symptom types is essential not only for clinicians seeking to provide appropriate care, but also for families, caregivers, and the general public striving to better understand this often misrepresented condition. At first glance, a patient experiencing positive symptoms of schizophrenia may appear entirely different from one displaying primarily negative symptoms. The contrast can be striking. One person might seem agitated, expressive, and caught in the grip of vivid hallucinations of fragmented, delusional beliefs, while the other may appear withdrawn, unresponsive, and emotionally flat, barely engaging with the world at all. Superficially, these two presentations could not be more different. One patient might speak rapidly about imagined conspiracies or claim to hear voices issuing commands. The other might sit in silence for hours, unable or unwilling to speak, their face unmoved by joy, sorrow, or even fear. To an untrained observer, it could seem as though these two individuals are suffering from entirely unrelated conditions, and yet, despite their outward dissimilarity, Both forms of schizophrenia arise from a common underlying disruption of brain function at the neurobiological level. Both positive and negative symptoms are linked to dysregulation in dopamine pathways, especially in regions of the brain responsible for perception, motivation, and executive function. In particular, overactivity in mesolimbic dopamine circuits is thought to contribute to positive symptoms, while underactivity in the mesocortical pathway may be associated with negative symptoms. Moreover, both patients often share cognitive impairments, such as difficulties with attention, working memory, and decision-making, deficits that may not be immediately visible but deeply affect day-to-day functioning, even when one symptom cluster is more prominent. The full spectrum of the illness often lies beneath the surface. This shared biological and cognitive architecture reminds us that schizophrenia is not a collection of isolated traits, but rather a single, complex disorder with diverse and dynamic expressions. Understanding this helps clinicians approach treatment with nuance and helps families recognize that, beneath outward differences, there is a shared and deeply human struggle occurring in both individuals. Schizophrenia has a strong genetic component, and one of its most intriguing and challenging features is how differently it can manifest, even among individuals who share similar genetic risk factors. The disorder often runs in families, and studies of twins, siblings, and parents have consistently shown that heredity plays a significant role in determining who is at risk. However, the expression of that risk is far from uniform. For example, two individuals who carry the same genetic variants associated with schizophrenia, even siblings raised in the same household, can present with vastly different symptom profiles. One might experience primarily positive symptoms, such as hallucinations, delusions, and disorganized thinking, while the other might display mostly negative symptoms, such as emotional flatness, withdrawal from social interaction, or a profound lack of motivation. This diversity in symptom presentation speaks to the complex interplay between genes and environment. It's not just which genes a person has, but how, when, and where those genes are expressed. A process influenced by factors like stress, trauma, substance use, hormonal changes, and even early childhood experiences. Furthermore, symptom expression can change over time within the same individual. A patient might experience one episode dominated by intense psychosis and disordered perception, while a later episode may be marked by profound apathy, emotional numbness, and a retreat from the world. This fluctuation suggests that schizophrenia is not a fixed state, but a dynamic condition that can evolve across the lifespan, shifting between different phases and expressions. This variability complicates diagnosis and treatment, but it also offers hope. It means that no single episode defines the entire course of the illness, and that with appropriate care, symptom patterns can shift and even improve. It also underscores the need for personalized treatment approaches, tailored not just to the diagnosis, but to the individual's unique history, biology, and current needs. In the end, understanding schizophrenia requires more than identifying its genetic roots. It calls for a holistic view, one that embraces its biological complexity, respects its psychological depth, and responds compassionately to its human face in all its many forms. The predominant type of symptoms a patient experiences, whether they are positive or negative, can offer important clues about how effective medication might be in their particular case. This is because the history of antipsychotic drug development is closely tied to one specific aspect of schizophrenia: the treatment of positive symptoms. In fact, the medications most commonly used today, particularly the first-generation antipsychotics, were not designed through an understanding of schizophrenia's full complexity. Instead, their discovery was serendipitous, emerging from early 20th century attempts to develop drugs for entirely different medical purposes. When certain compounds were found to reduce hallucinations, delusional thinking, and agitation in psychiatric patients, they were quickly repurposed as treatments for schizophrenia. These medications primarily target dopamine pathways in the brain, particularly the D2 dopamine receptors. Since positive symptoms are thought to be linked to an overactivity of dopamine in specific brain regions, such as the mesolimbic pathway, these drugs can be remarkably effective at reducing the intensity of hallucinations and delusions. and bringing a sense of external reality back into focus. However, this pharmacological approach has significant limitations, especially when it comes to the negative symptoms of schizophrenia, such as emotional blunting, social withdrawal, and cognitive dulling. These symptoms are often less responsive to current medications, and in some cases may even be exacerbated by them, particularly with long-term use of older antipsychotics as a result. Individuals who predominantly experience negative symptoms may find only modest or incomplete relief through drug therapy alone. This disparity highlights a fundamental issue in the treatment of schizophrenia. Most available drugs were not designed to address the full spectrum of the disorder. Instead, they emerged as solutions to the most visible and disruptive symptoms, leaving the more subtle, inwardly experienced struggles, often those that most impact quality of life, less effectively treated. Understanding this history is crucial for both clinicians and patients. It means that when evaluating treatment options, the balance of symptoms should guide not just medication choices, but also the integration of additional therapies, such as cognitive rehabilitation, social skills training, psychotherapy, and lifestyle support. In essence, the nature of a patient's symptoms helps shape the roadmap for care, pointing to where drugs may be most effective and where other interventions are essential. The discovery of antipsychotic medication marked a turning point in the history of psychiatry, especially in the treatment of individuals diagnosed with schizophrenia. Before these drugs emerged, many people suffering from the condition were confined for life in asylums or psychiatric institutions, often in harsh and dehumanizing conditions, with little hope for recovery or reintegration into society. But the introduction of the first antipsychotic medications in the 1950s, beginning with chlorpromazine, brought about a quiet revolution. For the first time, there was a pharmaceutical intervention that could meaningfully reduce the intensity of psychotic symptoms, such as hallucinations, delusions, and severe agitation. Patients who had once been unreachable in their delusions began to regain contact with reality, and the very concept of treatment began to shift from mere containment to active healing. This medical breakthrough catalyzed a broader transformation in mental health care as symptoms became more manageable through medication. The rigid model of permanent institutionalization began to give way. In its place emerged a more humane, multidimensional approach, combining psychotherapy, social rehabilitation, and pharmacological support. The focus moved toward community-based care, enabling many individuals to live outside hospital walls, rebuild relationships, pursue education or work, and participate in everyday life. Importantly, this transition also signaled a growing recognition of schizophrenia as a complex, treatable condition, not simply a life sentence or a reason for social exile. The use of antipsychotic drugs allowed other therapeutic modalities like cognitive behavioral therapy, family counseling, and social work interventions to become more effective, as patients were better able to engage and respond once their most acute symptoms were stabilized. Yet while the impact of these medications was profound, they were never a complete solution. Their discovery opened a door, but it also revealed the limitations of a purely medical model. True progress in treating schizophrenia would depend on weaving together science, compassion, and community, recognizing that recovery is not just about symptom control, but about restoring dignity, purpose, and human connection. Because antipsychotic medications often produce a striking reduction in the positive symptoms of schizophrenia, such as hallucinations, delusions, and disorganized thinking. Neuroscientists were inspired to develop theoretical models of the illness grounded in the observed effects of these drugs. The dramatic improvements seen in many patients following treatment suggested that the medications were not simply masking symptoms, but were acting on specific neurochemical pathways in the brain. This led researchers to ask a crucial question: if these drugs are effective, what does that tell us about the underlying biology of schizophrenia itself? In response, scientists began to reverse engineer their understanding of the disorder by examining how these medications work at the molecular and neural levels. Most antipsychotic drugs, particularly the early or typical antipsychotics, were found to exert their primary effects by blocking dopamine D2 receptors in the brain. Dopamine is a neurotransmitter involved in many cognitive and emotional functions, including reward, motivation, and perception. The success of dopamine-blocking medications led to what became known as the dopamine hypothesis of schizophrenia, a theory suggesting that an overactivity of dopamine signaling, especially in certain brain regions like the mesolimbic pathway, plays a central role in the emergence of psychotic symptoms. This hypothesis not only shaped decades of research but also provided a biological framework for understanding what had previously been seen as a mysterious and largely untreatable condition. It was one of the first times that a mental illness was linked directly to a specific neurochemical imbalance, helping to legitimize psychiatric disorders as biological diseases of the brain rather than purely psychological or moral failings. However, while the dopamine hypothesis offered a compelling explanation for the effectiveness of antipsychotics in treating positive symptoms, it soon became clear that this model could not fully account for the complexity of the disorder, particularly the negative and cognitive symptoms, which often responded poorly to dopamine-blocking drugs. Still, this early insight into the role of dopamine marked a milestone in the scientific study of schizophrenia, laying the groundwork for a more nuanced exploration of the brain's chemistry and the many factors, genetic, developmental, environmental, that contribute to this profoundly complex condition. Most antipsychotic medications work by binding to dopamine receptors in the brain, particularly the D2 subtype, and effectively blocking them from being activated by naturally occurring dopamine. In doing so, they reduce the overstimulation of neural pathways that are believed to be responsible for many of the hallmark symptoms of schizophrenia, such as hallucinations and delusions. This mechanism of action gave rise to the influential dopamine hypothesis of schizophrenia, which proposes that the disorder is primarily caused by an overactivity of dopamine transmission, especially involving D2 receptors. According to this theory, when too many D2 receptors are activated, particularly in the mesolimbic pathway, a brain circuit involved in emotion and reward, it leads to distorted perceptions of reality, impaired thinking, and other positive symptoms of schizophrenia. Supporting evidence came not only from the clinical effectiveness of antipsychotic drugs but also from brain imaging studies, which showed increased dopamine activity in certain regions of the brain in individuals experiencing psychotic episodes. Additionally, substances like amphetamine, which boost dopamine levels, can trigger psychosis in healthy individuals and worsen symptoms in people with schizophrenia, further reinforcing the connection between dopamine and the disorder. However, the dopamine hypothesis has its limitations. While it explains the positive symptoms relatively well, it falls short in accounting for negative symptoms such as emotional flatness, lack of motivation, and social withdrawal, as well as cognitive deficits like impaired attention and memory. These symptoms often persist even when dopamine activity is effectively blocked. As a result, researchers have come to view the dopamine hypothesis as a central piece of a much larger puzzle, one that includes glutamate dysfunction, neurodevelopmental abnormalities, and environmental stresses. Nonetheless, the insight that dopamine dysregulation, especially overactivation of D2 receptors, plays a key role in schizophrenia remains a foundational concept in modern psychiatry and pharmacology. Dopamine receptors are not isolated to a single part of the brain. They are found throughout various regions, each responsible for different functions such as emotion, motivation, decision-making, and movement. Because of this widespread presence, the dopamine hypothesis of schizophrenia evolved to become more nuanced, proposing that different symptoms of the disorder may be linked to dysfunction in distinct dopamine pathways. Specifically, this refined hypothesis suggests that the positive symptoms of schizophrenia, such as hallucinations, delusions, and disorganized thinking, are primarily caused by excess dopamine activity in the mesolimbic pathway. This pathway connects the ventral tegmental area (VTA) to regions like the nucleus accumbens and plays a key role in reward processing and emotional regulation. When dopamine signaling becomes hyperactive in this circuit, it can lead to a distorted sense of reality and inappropriate emotional responses. In contrast, the negative symptoms such as apathy, lack of emotional expression, and social withdrawal may be linked to dopamine underactivity in a separate circuit known as the mesocortical pathway. This pathway connects the VTA to the prefrontal cortex, an area involved in higher-order thinking, motivation, and social Behavior, a deficiency in dopamine transmission here is thought to contribute to the flattening of affect and cognitive impairments commonly observed in schizophrenia. Thus, the dopamine hypothesis doesn't propose a simple too much dopamine model, but rather a circuit-specific dysregulation, where some pathways are overactive and others are underactive. This dual imbalance could explain why a single disorder can produce such contrasting symptoms from chaotic hallucinations to lifeless emotional withdrawal. This understanding also helps explain why antipsychotic medications, which block dopamine receptors broadly, can successfully reduce positive symptoms but often have little to no effect on negative symptoms, and in some cases, may even worsen them. It highlights the complexity of treating schizophrenia and the need for targeted therapies that address multiple pathways rather than applying a single solution across the entire brain. It's also possible that individuals with schizophrenia may have an altered distribution or density of dopamine receptors across different regions of the brain. In other words, not only might certain dopamine pathways be overactive or underactive, but the physical arrangement and abundance of dopamine receptors themselves may differ from those in a neurotypical brain. This variation could contribute to the imbalanced signaling observed in the disorder, amplifying or dampening the effects of dopamine in critical neural circuits. Such differences in receptor distribution may be rooted in genetic factors. Dozens of genes have been associated with increased risk for schizophrenia, many of which play roles in brain development, synaptic function, and neurotransmitter regulation. Some of these genes may influence how many dopamine receptors are produced, where they are located, or how sensitive they are to dopamine, ultimately affecting the way the brain processes information, emotions, and perception. But genetics alone do not tell the whole story. It's increasingly clear that gene expression, the way genes are turned on or off, can be shaped by environmental influences. For example, early life trauma, chronic stress, or significant hormonal changes during adolescence may interact with a person's genetic makeup, activating or silencing certain genes at critical periods of brain development. This dynamic interplay between genes and environment is known as epigenetics, and it could explain why some individuals with genetic risk factors go on to develop schizophrenia while others do not. Adolescence, in particular, is a key window of vulnerability. During this time, the brain undergoes dramatic structural and chemical changes, including synaptic pruning and hormonal fluctuations. If a person carries certain genetic predispositions, these normal developmental shifts could potentially trigger the onset of schizophrenia, especially if compounded by psychosocial stresses or traumatic experiences. Therefore, the disease likely emerges from a complex web of biological and environmental factors, with dopamine receptor distribution being just one piece of a much larger puzzle. Understanding this complexity is essential for moving beyond a one-size-fits-all approach to treatment and toward more personalized, biologically informed interventions. So, the presence of symptoms in schizophrenia, and indeed in many mental health conditions, is not determined solely by how much of a neurotransmitter like dopamine is present in the brain. While the concentration of neurotransmitters plays an important role, it's only one part of the story. The number and sensitivity of receptors that these neurotransmitters bind to are equally crucial in shaping how brain signals are processed. Think of neurotransmitters as messages and receptors as the mailboxes that receive them. Even if there are plenty of messages being sent, if there are too many or too few mailboxes, or if the mailboxes are faulty, the communication system of the brain becomes disrupted. An imbalance in the number or function of receptors can lead to overstimulation or underactivation of key brain regions, giving rise to the wide range of symptoms seen in disorders like schizophrenia. Importantly, the quantity and distribution of receptors are not fixed. They can be built, modified, or even removed over time through a process influenced by gene expression. This means that even if someone inherits a certain genetic code, environmental factors can determine how those genes are turned on or off, thereby shaping the brain's architecture and chemistry. For example, chronic stress, early life trauma, substance use, or significant hormonal shifts, particularly those occurring during adolescence, can all affect receptor development and plasticity. Hormones like cortisol released during stress, or sex hormones like estrogen and testosterone, can also interact with genes and alter receptor density in various brain regions in sensitive individuals. This can tilt the brain toward dysregulated emotional responses, impaired cognition, or distorted perception, hallmarks of many psychiatric disorders. This means that mental illness cannot be explained simply by too much or too little of a chemical. It's about how those chemicals are received, interpreted, and modulated by the brain's complex receptor systems, which are themselves shaped by a lifetime of biological and experiential factors. Understanding this dynamic opens the door to more nuanced and personalized approaches to mental health treatment, moving beyond chemical imbalance models and toward a deeper understanding of brain plasticity and individual neurobiology. But here's where the chemical imbalance theory begins to unravel, especially when applied to complex conditions like schizophrenia. The idea that simply having too much dopamine causes mental illness is overly simplistic and increasingly outdated. If it were that simple, treatment would be straightforward: just reduce dopamine levels in the brain and the symptoms would disappear. But that's not what we see in reality. It's not just the amount of dopamine that matters, but where in the brain it's acting and how it's interacting with specific types of dopamine receptors. Schizophrenia, for example, has been linked to overactivation of dopamine D2 receptors in certain subcortical areas of the brain, like the striatum, which may contribute to hallucinations or delusions, what we call positive symptoms. But here's the catch: the same dopamine-blocking drugs that reduce these positive symptoms often fail to address the negative symptoms of schizophrenia, such as emotional flatness, social withdrawal, or lack of motivation. In some cases, they may even worsen them. These negative symptoms are believed to be associated with reduced dopamine activity in other brain regions, like the prefrontal cortex, suggesting a regional imbalance, not a global excess. This complexity puts proponents of the old chemical imbalance narrative in a difficult position. If schizophrenia was simply a matter of too much dopamine, blocking dopamine receptors should alleviate all symptoms, but that's not what happens. In fact, many antipsychotic medications, particularly the older, typical ones, primarily target dopamine but have little to no effect on the negative or cognitive symptoms, which are often the most disabling for patients. Furthermore, newer research shows that dopamine is only one part of a much larger, more intricate web of interacting neurotransmitters and neural circuits: glutamate, GABA, serotonin, and even inflammatory markers and stress hormones may all play critical roles. Schizophrenia appears to be not a chemical imbalance, but a network disorder, a disruption in how various parts of the brain communicate, adapt, and regulate themselves. So while dopamine does matter, it's where, how, and in what context it's acting that shapes the outcome, not simply how much there is. This challenges the outdated notion that mental illness can be treated like a plumbing problem where we just open or close the chemical valves. Instead, it demands a more holistic, individualized understanding of the brain, one that considers genetics, trauma, development, and neuroplasticity alongside chemistry. Still, despite its limitations, the dopamine hypothesis of schizophrenia retains support from several compelling lines of evidence, particularly in relation to the disorder's positive symptoms, such as hallucinations and delusions. While it's clear that the hypothesis cannot explain the full spectrum of schizophrenia, certain findings do continue to reinforce its relevance, especially when viewed through the lens of pharmacology and neuroscience. One such source of support comes from observations involving dopamine-enhancing drugs, especially psychostimulants like amphetamines or cocaine. These substances work by increasing the availability of dopamine in the brain, often by promoting its release or blocking its reuptake. When used in high doses or over extended periods, these drugs can induce psychotic symptoms strikingly similar to those observed in schizophrenia, including paranoia, auditory hallucinations, and delusional thinking. This phenomenon, stimulant-induced psychosis, has been well documented and serves as a powerful clue. If artificially elevating dopamine levels can cause psychosis in otherwise healthy individuals, it suggests that abnormal dopamine signaling could be a key factor in triggering these symptoms in vulnerable populations. In fact, this has led some researchers to propose that stimulant-induced psychosis may serve as a model for understanding the dopamine-related mechanisms of schizophrenia, particularly those underlying its most overt and disruptive manifestations. Moreover, Imaging studies have shown that individuals experiencing psychosis, whether drug-induced or related to schizophrenia, often exhibit increased dopamine synthesis and release in subcortical areas of the brain, such as the striatum. These findings provide a biological anchor to the behavioral observations, supporting the notion that excessive dopamine activity in specific regions plays a significant role in the emergence of psychotic symptoms. However, it's important to note that this doesn't prove dopamine is the root cause of schizophrenia, nor does it suggest that all cases of psychosis share the same underlying biology. But it does add weight to the idea that dopamine dysregulation is a significant contributor, especially in the acute, florid phases of the disorder. In this sense, the dopamine hypothesis still holds partial validity, not as a complete explanation, but as one piece of a much larger and more intricate puzzle. It helps illuminate why certain medications work as they do, and why particular symptoms emerge under specific neurochemical conditions. The challenge for modern neuroscience is not to discard this theory entirely, but to integrate it with a broader, multidimensional understanding of schizophrenia, one that includes genetics, brain connectivity, immune factors, early developmental influences, and the role of trauma. Given this body of evidence, it's easy to see why the idea that schizophrenia is caused by too much dopamine has held such appeal for decades. The connection between elevated dopamine activity and the manifestation of psychotic symptoms appears at first glance both logical and compelling. After all, medications that reduce dopamine signaling often alleviate symptoms like hallucinations and delusions, while drugs that increase dopamine levels can produce those very symptoms in otherwise healthy individuals. However, this interpretation is an oversimplification of a much more complex reality. The dopamine hypothesis, first proposed in the mid-20th century, was never just about the notion that more dopamine equals schizophrenia. Rather, it suggested that dysregulation of dopamine pathways, particularly hyperactivity in specific regions of the brain such as the mesolimbic system, might underlie certain symptoms of the disorder. Over time, though, our understanding of schizophrenia has grown more sophisticated, and so too has the framework of the dopamine hypothesis. Researchers now recognize that dopamine's role in the illness is highly region-specific, with excess dopamine activity in some brain areas possibly coexisting with deficits in others. For instance, while increased dopamine transmission in the striatum may drive positive symptoms like delusions and hallucinations, reduced dopamine activity in the prefrontal cortex has been linked to negative symptoms and cognitive impairments such as flattened affect, poor executive function, and difficulty concentrating. More importantly, a growing body of emerging research challenges the idea that dopamine is the central or primary cause of schizophrenia. Although dopamine clearly plays a role, particularly in the pharmacological treatment of symptoms, it may not be the originating factor in the disorder's development. Rather, it might represent a downstream effect or one component of a larger cascade of neurobiological changes. Other systems are now under close investigation, especially the glutamate system, which governs excitatory signaling in the brain. Abnormalities in glutamatergic transmission, particularly involving the NMDA receptor, have been found to produce symptoms that closely resemble those of schizophrenia, even in the absence of dopaminergic disturbance. This has led to the proposal of the glutamate hypothesis, which may offer a more comprehensive account of the disorder's wide-ranging symptoms, including those that dopamine-focused theories struggle to explain. Additionally, newer models suggest that schizophrenia may result from disruptions in the brain's broader neural networks, involving complex interactions between dopamine, glutamate, GABA, and other neurochemical systems, as well as factors like neuroinflammation, oxidative stress, synaptic pruning errors, and early developmental insults. In short, while the dopamine hypothesis has contributed immensely to our understanding and treatment of schizophrenia, it is no longer seen as the complete picture. Instead, it has evolved into part of a multidimensional framework where dopamine plays a crucial but not exclusive role in a far more interconnected and dynamic neurobiological landscape. One important challenge to the traditional dopamine hypothesis is that many individuals with schizophrenia do not respond adequately to medications that specifically block D2 dopamine receptors. This inconsistency has prompted researchers to explore alternative explanations and refine their understanding of the disorder. Additional insight has come from the development and use of atypical antipsychotic medications, a newer generation of drugs that tend to produce fewer motor-related side effects and are often better tolerated by patients, unlike the older, typical antipsychotics, which act primarily by strongly blocking D2 receptors. Atypical antipsychotics are less potent in their dopamine-blocking capacity, yet they still effectively reduce positive symptoms such as hallucinations and delusions. Interestingly, they are also more successful at improving negative symptoms like emotional flatness, social withdrawal, and lack of motivation, areas where traditional medications often fail. This discrepancy suggests that the therapeutic action of atypical antipsychotics may not rely solely on dopamine modulation. In fact, these medications also interact with other neurotransmitter systems, most notably serotonin, by targeting both dopamine and serotonin receptors. Atypical antipsychotics point to a more complex neurochemical imbalance in schizophrenia than originally thought, expanding the scope of inquiry beyond a single neurotransmitter. In addition to dopamine and serotonin, another major neurotransmitter, glutamate, has come under increasing scrutiny in the search for a more comprehensive understanding of schizophrenia. Glutamate is the brain's primary excitatory neurotransmitter, playing a vital role in cognitive functions such as learning, memory, attention, and neuroplasticity. Emerging evidence suggests that dysregulation in the glutamatergic system may contribute significantly to the development and expression of schizophrenia symptoms. Support for this theory comes from studies involving drugs that block glutamate receptors, particularly the NMDA and methyldiaspartate subtype, substances like phencyclidine PCP and ketamine, which act as NMDA receptor antagonists, can induce a range of schizophrenia-like symptoms, including hallucinations, delusional thinking, and cognitive impairments even in individuals with no psychiatric history. This has led researchers to consider whether a deficiency in NMDA receptor function might be a core feature of the disorder. Interestingly, nicotine, a substance with well-documented effects on the brain's reward and attention systems, has also been linked to glutamate regulation. Nicotine stimulates receptors that enhance the release of glutamate in the prefrontal cortex, a brain region critically involved in decision-making, attention, and executive function. This may help explain why a disproportionately high percentage of people with schizophrenia smoke cigarettes. Many patients report that nicotine improves their focus, reduces cognitive fog, and alleviates some of the negative symptoms that are otherwise difficult to treat with medication. Taken together, these findings suggest that schizophrenia is likely not the result of a single neurotransmitter imbalance, but rather a complex interplay between multiple neural systems, with glutamate playing a potentially central yet underappreciated role in the disorder's cognitive and perceptual disturbances. In recent years, neuroscientists have proposed what is known as the glutamate hypothesis of schizophrenia, offering a more integrative perspective on the disorder's underlying neurochemical imbalances. This theory suggests that glutamate, the brain's primary excitatory neurotransmitter, plays a critical regulatory role in maintaining the balance of other neurotransmitter systems, particularly dopamine. Under normal conditions, glutamate activity helps modulate and restrain dopamine signaling within key brain circuits, especially those involved in cognition, emotion, and perception. However, in individuals with schizophrenia, it is believed that there is a deficiency or dysfunction in glutamatergic transmission, particularly through NMDA receptors. This glutamate shortfall results in a loss of inhibitory control over dopamine, allowing dopamine activity to become dysregulated and overactive in certain brain regions. This imbalance may help explain the dual nature of schizophrenia's symptoms. Positive symptoms, such as hallucinations and delusions, which are linked to excessive dopamine activity in subcortical regions like the mesolimbic pathway, and negative symptoms, such as emotional flatness and social withdrawal, which may be tied to diminished glutamate function in the prefrontal cortex. In this view, schizophrenia is not solely a dopamine disorder, but rather the result of a cascade effect where insufficient glutamate signaling disrupts the brain's delicate neurochemical equilibrium, leading to widespread functional impairments. This hypothesis helps to bridge the gap between older dopamine-based models and the growing body of evidence pointing to more complex, multi-system dysfunctions in the brain. However, Attempts to treat schizophrenia by directly increasing glutamate levels in the brain have yielded disappointing results, particularly when it comes to reducing the positive symptoms, such as hallucinations and delusions, while the glutamate hypothesis suggests that a deficiency in glutamatergic signaling plays a role in the disorder. Simply, boosting glutamate levels does not appear to reverse the overactivity of dopamine that underlies these more dramatic symptoms. One reason for this limited success may be that glutamate functions in highly complex and region-specific ways, and its role in the brain involves intricate feedback loops and interactions with other neurotransmitters. Increasing glutamate indiscriminately across the brain can lead to neurotoxicity or overstimulation, potentially causing more harm than benefit. Moreover, positive symptoms are thought to arise primarily from hyperactivity in the mesolimbic dopamine system, which may not be adequately modulated through glutamate-based interventions alone. As a result, while glutamate remains a critical piece of the puzzle in understanding schizophrenia, therapeutic strategies targeting this system are still in early development, and effective treatments may require a more targeted and nuanced approach, perhaps focusing on specific glutamate receptors such as NMDA or mGlu subtypes, rather than attempting to elevate glutamate levels globally. And to make the picture even more complex, it's not just chemical imbalances such as neurotransmitter levels that distinguish the brains of individuals with schizophrenia from those of neurotypical individuals. Structural differences in the brain have also been consistently observed in people with the disorder. Neuroimaging studies have revealed that certain brain regions tend to show reduced volume in many patients with schizophrenia, notably the frontal cortex, which is crucial for decision-making, planning, impulse control, and social behavior, often appears smaller. Likewise, the temporal lobes, which play key roles in language processing, auditory perception, and memory, also show signs of atrophy or underdevelopment in affected individuals. These physical differences may help explain some of the cognitive and emotional challenges associated with schizophrenia. For example, reduced volume in the prefrontal cortex is linked to difficulties with executive functioning such as organizing thoughts, maintaining attention, and regulating behavior. Abnormalities in the temporal lobes, particularly in the hippocampus and amygdala, may contribute to distorted perceptions, memory disruptions, and emotional dysregulation, all of which are hallmarks of the illness. Importantly, these structural differences are not static. Some studies suggest that brain volume loss may progress over time, particularly in the early stages of schizophrenia, which raises questions about whether the illness causes gradual degeneration or whether these changes are present even before symptoms emerge. This line of research underscores the likelihood that schizophrenia is not solely a disorder of neurotransmitters or thoughts. It's also a disorder of brain development and organization, influenced by a combination of genetics, environment, and early life experiences, in addition to structural differences. Researchers have also identified functional abnormalities in the brains of people with schizophrenia. These refer not to the physical size or shape of brain regions, but to how those regions activate and communicate during specific tasks or resting states. Functional imaging studies, such as fMRI and PET scans, have revealed that areas like the frontal cortex, temporal lobes, and hippocampus often show atypical patterns of activity in individuals with schizophrenia. For example, the frontal cortex, which is typically engaged during problem-solving, reasoning, and decision-making, may show reduced activation during tasks that require executive control. This may help explain the difficulties with attention, working memory, and goal-directed behavior often seen in schizophrenia. Similarly, the temporal lobes, involved in processing language and auditory information, may respond abnormally to sound or speech, potentially contributing to auditory hallucinations. One of the hallmark symptoms of the disorder, the hippocampus, a region essential for memory formation and spatial navigation, may also function inefficiently, leading to confusion, disorganized thinking, or impaired memory. Beyond individual regions, the connectivity between brain areas, the way they coordinate and share information, also appears disrupted in healthy individuals. Different parts of the brain work in a synchronized and balanced way, forming networks that underlie cognition, emotion, and perception. In schizophrenia, however, these networks often show abnormal connectivity. Some connections may be weakened, leading to fragmentation of thought or loss of coherence, while others may be excessively strong or misrouted, contributing to distorted perceptions and delusions. This altered brain communication suggests that schizophrenia may not just be a problem within specific brain regions, but a disorder of network integration, a kind of neurological miswiring that affects how the brain functions as a whole. Understanding these functional and connective differences is key to developing better treatments and perhaps even early detection strategies for the illness. Despite the growing body of evidence documenting both structural and functional abnormalities in the brains of individuals with schizophrenia, the root cause of these changes remains uncertain. One of the most significant challenges in schizophrenia research is disentangling cause from consequence. Are these brain differences driving the onset of the illness, or are they the result of having the disorder over time? Or perhaps are they at least partially caused by long-term use of antipsychotic medications themselves? For instance, some neuroimaging studies have observed progressive reductions in grey matter volume in certain brain regions over the course of the illness. But whether this represents a natural trajectory of the disease or a side effect of chronic medication use is still under debate. Similarly, functional differences in how the brain activates or connects may emerge before symptoms appear, suggesting a possible causal role, yet they could also develop later as a byproduct of psychosis, stress, or social withdrawal. Moreover, the question of what underlies the observed neurotransmitter imbalances, such as dysregulation of dopamine or glutamate systems, remains largely unanswered. We know that these chemical messengers function abnormally in many patients, but we do not yet understand why this occurs. Are genetic mutations to blame? Environmental stresses? Prenatal complications? Or some complex interaction among all three? In essence, schizophrenia presents a chicken or egg dilemma at nearly every level of analysis. Are the brain abnormalities and neurotransmitter changes the drivers of the disorder, the consequences, or perhaps both, in a self-reinforcing cycle? Until we can answer these questions, our understanding remains incomplete, and treatment continues to be focused more on managing symptoms than curing the root cause. Depression. The widely circulated notion that depression is simply the result of a chemical imbalance in the brain, most often involving serotonin, is a persistent and oversimplified myth. This idea, though convenient and easy to market, does not accurately reflect the complex reality of how depression works. The origins of the chemical imbalance theory can be traced back to the accidental discovery of early antidepressants in the 1950s. Such as imipramine, a tricyclic antidepressant. Researchers noticed that this drug, originally tested for other conditions, seemed to improve mood in certain patients. Later studies showed that it increased levels of neurotransmitters like serotonin and norepinephrine, which led to the hypothesis that a deficiency in these chemicals might cause depression. This hypothesis became the foundation for the development of selective serotonin reuptake inhibitors (SRIs), such as Prozac, and was heavily promoted by pharmaceutical companies. The result was a powerful narrative: depression is caused by a lack of serotonin, and SRIs fix it. But this view is outdated and scientifically incomplete. In reality, depression is far more multifaceted. It involves not only chemical messengers, but also neural circuitry, genetics, inflammation, hormonal dysregulation, and the person's psychosocial environment. It's also important to note that not all people with low serotonin levels experience depression, and not all people with depression show signs of serotonin deficiency. In fact, some antidepressants work without altering serotonin levels significantly at all. Thus, while the chemical imbalance theory helped ignite early pharmacological treatment of depression, it no longer holds up as a comprehensive explanation. It's a partial view at best and a misleading one at worst if it causes people to believe their distress is due solely to a broken brain chemistry that can be corrected with a pill. Today, we understand that tricyclic antidepressants (TCAs) exert their effects primarily by targeting a group of neurotransmitters known as monoamines, which include dopamine, serotonin, and norepinephrine. These chemical messengers play a crucial role in regulating mood, attention, motivation, and emotional stability. Tricyclics work by inhibiting the reuptake of these neurotransmitters in the brain, meaning they prevent the chemicals from being reabsorbed back into the nerve cells that release them. This leads to increased levels of these monoamines in the synaptic cleft, the small space between neurons, making them more available to bind with receptors on neighboring neurons by enhancing the transmission of these mood-related signals. Tricyclics were found to alleviate symptoms of depression in many patients. However, the mechanism is not entirely straightforward, and the therapeutic effects typically take weeks to emerge, even though the chemical changes occur within hours. This time lag suggests that secondary processes such as changes in gene expression, neuroplasticity, or receptor sensitivity may also play significant roles in recovery. While this discovery supported the monoamine hypothesis of depression, it also opened the door to decades of debate and research, revealing just how intricate and interconnected the neurobiology of mood disorders truly is. Around the same time that imipramine was gaining attention for its surprising antidepressant effects, another serendipitous discovery emerged, this time from a very different corner of medicine. A group of physicians treating tuberculosis patients observed an unexpected side effect, a noticeable elevation in mood, sometimes bordering on euphoria. The drug in question, originally developed to fight tubercular infection, turned out to have profound effects on brain chemistry. It was later identified as a monoamine oxidase inhibitor, a class of compounds that target the enzyme monoamine oxidase, which is responsible for breaking down key neurotransmitters like serotonin, norepinephrine, and dopamine. By inhibiting this enzyme, the drug effectively slowed the degradation of these neurotransmitters, allowing them to accumulate and remain active longer in the synapses between neurons. As a result, more monoamines were available to influence mood-regulating circuits in the brain, leading to the improved emotional states observed in patients. This unexpected psychiatric benefit helped solidify the idea that depression might be linked to deficits in monoamine activity. The logic was compelling in its simplicity. If increasing serotonin and norepinephrine levels improves mood, perhaps low levels of these chemicals are what caused depression in the first place. Thus, the monoamine hypothesis of depression was born not out of theoretical deduction, but from a series of fortunate pharmaceutical accidents. And while this hypothesis would dominate psychiatric thinking for decades, it also sparked a wave of new treatments and theories, each seeking to better explain and treat the complex nature of mood disorders. Many modern antidepressant medications target the brain's monoamine neurotransmitters, primarily norepinephrine, dopamine, and serotonin, but while these chemicals often get grouped together, each plays a distinct and nuanced role in regulating our thoughts, emotions, and behaviors. Norepinephrine acts like the brain's internal amplifier, enhancing alertness, attention, and overall arousal. It prepares the mind to respond to challenges, sharpen focus, and maintain vigilance. In a depressed brain, where motivation and mental clarity may be blunted, increasing norepinephrine levels can sometimes help lift the mental fog. Dopamine, on the other hand, is often described as the neurochemical of reward. It fuels motivation, pleasure, and the ability to experience joy and anticipation. It's also involved in working memory, the mental space where we hold and manipulate information. When dopamine is dysregulated, people may struggle with apathy, emotional flatness, or a lack of drive. Serotonin serves yet another purpose. It is more subtly involved in maintaining emotional balance. Elevated serotonin levels can dampen anxiety, reduce obsessive thought patterns, and ease compulsive behaviors. In this way, it promotes a sense of inner calm and emotional steadiness, a counterbalance to the agitation and repetitive rumination often found in depression. Though these neurotransmitters are often discussed as if they were interchangeable mood enhancers, the reality is far more complex. Each touches a different domain of experience, and altering their levels can produce very different effects, depending on the individual and the nature of their symptoms. Each of these neurotransmitter functions: attention, motivation, pleasure, memory, anxiety regulation is often disrupted in depression, though the extent and pattern of disruption can vary dramatically from one person to another. Depression is not a single, uniform condition. It is a complex constellation of symptoms that may manifest as deep emotional despair in one individual, paralyzing apathy in another, or relentless anxiety and rumination in a third. Because of this variability, modern psychiatry increasingly recognizes that there is no one-size-fits-all approach to treatment, rather than relying on a standard medication for all cases. Psychiatrists are trained to tailor pharmacological interventions, crafting a kind of chemical recipe that aligns with the unique symptom profile of each patient. A person struggling with profound fatigue and an inability to feel pleasure may benefit from drugs that enhance dopamine transmission. Someone overwhelmed by anxious thoughts and compulsions might respond better to medications that boost serotonin. For others, whose depressive fog is accompanied by poor concentration and lack of alertness, a norepinephrine-focused approach may be more effective. In this way, treating depression becomes both a science and an art, a delicate balancing act of understanding brain chemistry while attuning to the subjective, deeply personal nature of emotional suffering. If depression were merely the result of a simple chemical imbalance, then correcting that imbalance should bring about immediate relief. One might expect that once serotonin or dopamine levels are adjusted by medication, the clouds would lift and clarity would return, but that's not what happens with most antidepressant drugs. There's a well-documented phenomenon known as the therapeutic lag. The biochemical effects of the medication, such as increased levels of serotonin or norepinephrine, occur within hours or days of taking the drug. Yet, the emotional relief, the actual lifting of depressive symptoms, often doesn't arrive for weeks. This puzzling delay reveals something critical. Depression is not simply about missing chemicals. If it were, relief would be as swift as the neurochemical shift, but the brain is not a machine that responds immediately to a new input. It is a dynamic, adaptive organ. The delayed therapeutic response suggests that longer-term changes must be taking place, perhaps in neuroplasticity, gene expression, or the rewiring of emotional circuits. In other words, medication may lay the groundwork, but healing unfolds more slowly as the brain relearns how to process emotion, restore resilience, and rebuild its inner architecture of hope. What's even more revealing is that patients with depression often don't have low levels of monoamines to begin with. Despite the popularity of the chemical imbalance theory, research has shown that many individuals suffering from major depression have neurotransmitter levels in their cerebrospinal fluid that are indistinguishable from those of people without depression. Even more striking, artificially lowering monoamine levels in otherwise healthy individuals does not reliably induce depression. The mood does not collapse simply because serotonin drops, nor does joy vanish when dopamine is reduced. This suggests that the roots of depression are far more intricate than a matter of chemical scarcity. Neurotransmitters may play a role in the emotional landscape, but they are not the whole terrain. Depression may not be a shortage, it may be a misfiring, a disconnection, a disruption in how the brain processes meaning, memory, and emotion over time. It is not just about levels, it is about how the brain uses those chemicals, where it sends them, and what stories it constructs in the silence between signals. And then there's this: some patients experience significant relief from antidepressants that don't even target monoamines, at least not directly. These drugs seem to bypass the traditional chemical narrative altogether, yet still bring light to a mind dimmed by despair. This alone challenges the notion that depression is simply a matter of serotonin or dopamine gone awry. Even more compelling is the evidence from psychotherapy, especially cognitive behavioral therapy (CBT). In CBT, no chemicals are administered, no neurotransmitters are directly altered. Instead, a patient learns to notice, challenge, and reshape the thoughts that feed their suffering, and this process, in many cases, can be just as effective as pharmacological treatment, particularly for mild to moderate depression. But here's what's truly remarkable: these psychological interventions don't just change the mind, they change the brain. Brain scans reveal that CBT can produce neuroanatomical and physiological changes strikingly similar to those observed in patients taking antidepressants, as if the brain responds not only to molecules, but also to meaning, as if the act of seeing one's pain through a new lens is itself a biochemical event. In this light, depression begins to look less like a fault in our chemistry and more like a conversation between biology, experience, memory, and thought. Perhaps the final blow to the chemical imbalance theory comes from one of the most humbling findings in modern psychiatry. Many patients with mild to moderate depression improve just as much on a placebo as they do on active medication. A sugar pill, devoid of any pharmacological effect, can still lift the fog of despair if the patient believes it will. This isn't merely a testament to the power of belief, it's a revelation about the mind's ability to respond to expectation. Trust and hope. The placebo doesn't change brain chemistry in the traditional sense, but the very act of believing in healing can initiate a cascade of real, measurable physiological change. In this sense, the placebo effect is not a trick. It's a mirror reflecting a truth we've long tried to ignore. The brain is not a machine waiting to be fixed, but a dynamic system constantly shaped by meaning, context, and experience. So when a person begins to heal not because of what's in the pill, but because of what the pill represents, we are reminded that depression cannot be reduced to a formula. It speaks a language far more complex than serotonin, just like in schizophrenia. Depression leaves traces in the architecture of the brain, not just in chemicals, but in folds and circuits, in the slow reshaping of neural terrain. MRI scans and post-mortem studies reveal a landscape altered, hippocampi that shrink, prefrontal cortices that dim, amygdalae that flare like alarm bells, but perhaps more telling is the way the brain responds or fails to respond to experience. This is the realm of neuroplasticity, the brain's quiet ability to bend, to learn, to heal. In many who suffer from depression, this plasticity falters. The brain becomes less like clay, more like stone. In a world constantly shifting, the depressed brain struggles to adapt. It holds on to pain like a familiar language, resists change like a house where every door has been locked from the inside. And yet, even this is not universal. Not every brain changes the same way. Not every mind sinks to the same depth. But what is clear is this: depression is not simply a matter of sadness, it is a reshaping of self. In the evolving landscape of mental health, a new path emerges, one paved not just with chemicals, but with code. Genetic information now whispers secrets to psychiatrists. Which drug might fit which person? Like a key to a complex inner lock, it's the promise of precision, of tailoring the medicine to the soul's unique biology. And yet, we must not forget the mind behind the map. Beyond the double helix, beyond blood tests and receptor sites, there is story, there is history, there is the quiet power of words. Psychological therapy, conversation, reflection, reframing can also rewire the brain, not with pills, but with presence. New thoughts carve new neural paths, new beliefs rebuild broken bridges. In sum, it is this gentle reshaping, not pharmacology, that lifts the fog. The future, perhaps, lies in integration, biology and biography, gene and journey, molecule and meaning, because to treat the mind is not just to fix a machine, it is to understand a human being. For those who wish to journey deeper into the science and stories behind these ideas, there are several illuminating works worth exploring. J. Ban reflects on the curious role of serendipity in shaping drug discovery, offering insight into how chance has often guided the course of psychiatric medicine. In a revealing study, Barden, Esler, Dorward, Lambert, and colleagues examine the phenomenon of elevated brain serotonin turnover in patients with depression, adding complexity to the chemical narrative. Meanwhile, Dewall, Galath, Pressman, and Black explore a paradox in their work, uncovering how the so-called love hormone can, under certain conditions, lead not to peace, but to violence. And Lacasse and Leo provide a critical examination of the serotonin hypothesis itself, inviting readers to question long-held assumptions about depression and its treatment.